Happy Halloween.
Happy Halloween.
Today is about the reticular formation. This is a collection of nuclei within the brainstem that can create local circuits to carry out reflexive behaviors. They have ascending projections that can affect the thalamus and cortex to affect our wakefulness. That's going to be the second part of this. They also have descending outputs. We have the reticulospinal tract, for example. But more relevant to today would be the descending outputs that are going to control respiration and heart rate as well. So they can do it all. They're going to be located from the midbrain down to the medulla. So there's a variety of nuclei within the reticular formation. Those that are near the top tend to project upward. Those that are near the bottom tend to project downward. And those within the pons tend to stay local. They're going to hit the brain stem. That's your broad distinctions there. The more lateral regions are going to contain the small parvocellular neurons. They'll receive sensory input and project their axons a little more locally than the more central magnocellular neurons that are going to project over long distances, either upward or downward. The focus of today is on respiration, sleep-wake. So when we're talking about respiration and blood flow, this comes back to cranial nerves 9 and 10. Breathing is probably one of the most important things that we do, aside from, I guess, everything else, but you have to do it to stay alive. And it's one of the last things you'll do in this life. Immediately afterwards, you'll shit your pants. we lighten things up. So we want to be breathing all day, every day, but you don't want to necessarily have to think about it. Wouldn't it be nice if we had some reflexive circuits that could sense when we need to breathe and just carry that out for us? Well, luckily we do. We can sense blood pH as a proxy for carbon dioxide levels. Have we heard of the bicarbonate buffer in our blood? Excellent. When carbon dioxide increases, you make carbonic acid. Yeah, carbonic acid is going to dissociate into its bicarbonate base form and a proton, as long as the pH is above 3, which it always is. So when you have an increase in carbon dioxide, you're going to have an increase in acid. You'll have a drop in pH. That's what we're going to sense. We don't sense carbon dioxide because that freely flows through membranes. It's hard to have that bind to a receptor. But protons, on the other hand, despite being incredibly small, it's not the size here that matters, it's the charge. They have a positive charge. They can't flow through membranes. So we can detect that floating around in the blood, and that's what we're going to sense. We're going to sense the pH of our blood, and that's what's going to determine the rate of respiration. Because the whole purpose of respiration is to make sure that we don't have a buildup of carbon dioxide or a deficiency in oxygen. Nothing in life is free. And the way that we pay the bill in our cells is through ATP hydrolysis. So we have to always be making ATP, and that's of course dependent upon oxygen. So you're always using up oxygen and creating carbon dioxide. That's why we exhale. Since you've used up that oxygen, you need to inhale and bring in a fresh batch. We'll pump it around and keep the whole system working. When you don't have enough oxygen available, when there's a buildup of carbon dioxide, we see a drop in pH. When there's a drop in pH, this stimulates chemoreceptors that are going to be located peripherally out there on the carotid. So these will stimulate cranial nerve 9. Also centrally within the medulla. We're going to monitor the pH of the CSF and the blood supply there. By we, I mean the local astrocytes in the medulla. They have pH-sensitive potassium channels that are going to help them determine, are we getting a little acidic? Do we need to take a breath? It's the same mechanism whether you're in the central nervous system or out there at the carotid. A drop in pH is going to affect our potassium conductance. So we have pH-sensitive potassium channels that will allow this type of behavior, where a drop in pH, because they've given it a burst of carbon dioxide, so these gray lines here are showing us where they've applied high carbon dioxide. X is time, This Y is showing you the firing rate of those pH-sensitive neurons that are getting a little dose of ATP, as we'll soon find out. And this is because of the drop in pH. Small drops in pH are going to get picked up. Neurons then respond to that, they become active, and they stimulate inspiration. The way that this is directly measured is through pH-sensitive potassium channels. Here we're looking at our potassium conductance on the Y and the pH on the X. Now there's a few different pH-sensitive potassium channels. They have subtle differences in their conductance across different pHs, but you can see the same pattern here. Increase the pH, increase the potassium conductance. If we increase our potassium conductance, is that going to depolarize us or hyperpolarize us? Everything is normal now. We're not in the endolymph anymore. So is it going to depolarize or hyperpolarize? Hyperpolarize, exactly. So at high pH, they'll hyperpolarize. What we're concerned with is that low pH. Whenever you have a buildup of carbon dioxide, drop in pH, drop in potassium conductance. You move away from potassium's reversal, and that's why you see this increase in firing rate. The direct sensors of this would be the glomus cells and the astrocytes. Out there, the carotid, in the medulla. Of course, astrocytes aren't out there in the carotid, so we have the glomus cells here. Both of these have pH-sensitive potassium channels. They're going to depolarize whenever there's a drop in pH. When they depolarize, they start to spit out neurotransmitters. There's a variety of them that they'll spit out. The one that we'll talk about here is ATP. Not only do we spend ATP to pay the bill, we also use it as a neurotransmitter. When ATP is spat out, it's going to bind to these purinergic receptors here, P2X2 receptors. These are cation channels. If it's a cation channel, it's going to excite or inhibit the neurons. Cations. Excite, excellent. Yeah. Non-specific cation channels reverse around 0 millivolts. That's well above threshold. So these will excite the nearby neurons. Either cranial nerve 9, if we're out in the carotid, or those local medullary chemoreceptive neurons in the medulla. So there's a drop in pH, decrease in potassium conductance, we spit out ATP. That's going to bind to our P2X2 receptors, which are also, to some degree, pH dependent. The degree to which they bind ATP and conduct cations is dependent on pH. When pH drops, they bind ATP more readily and give you a stronger cation conductance. So the drop in pH is going to allow for excitation in both types of cells. And then the actual chemoreceptor over here that's a little more directly in contact with the blood spits out ATP to further excite the neurons. So when there's a drop in pH, we need to take a breath. A drop in pH is going to stimulate cranial nerve 9. There's also some local chemoreceptive neurons in the medulla. And these are all going to feed in to the solitary tract nucleus. Solitary tract, sensory. We broke it up into 4 nuclei before. One of those was the dorsal respiratory group. It's back. We're finally going to see what this thing does. So cranial nerve 9 out there in the carotid, or local afferents that aren't shown here, feed in and excite the dorsal respiratory group anytime we have a drop in pH. Now, remember there's also input from the lung afferents to sense stretch, so we know where we're at in this cycle. So based on whether there's stretch in our lungs or not, our dorsal respiratory group is going to know whether we're currently inhaling or exhaling, because that's going to determine the behavior as well. If there's a drop in pH, we know we want to inhale. If the lungs are already stretched, exhale first, then inhale. The dorsal respiratory group is going to have a couple different projections. It'll project downward to the inspiratory neurons. Down in the spinal cord, they're going to control our diaphragm and our intercostals. So they can stimulate those. They're also going to affect the timing mechanism, so the pre-Betzinger complex, one of those central pattern generators. They're going to project also to the ventral respiratory group which we divide up. Part of it's the prepensing complex. There's a rostral and a caudal portion. The caudal portion is going to allow for expiration. And then the pontine respiratory group. That's another central pattern generator that's going to allow us to control the respiration rate with other activities, like speaking. During those periods of time where I'm spouting out all those words at a rapid pace, I am not inhaling. So I need to suppress my desire to inhale, even if there's a slight drop in pH. The pons is going to work all that out. If we're chewing, swallowing, that's going to affect respiration as well. So the pontine respiratory group is going to control all of the rest of this lower stuff in the medulla based on what else we're doing, so that we don't inhale while swallowing, and then it chokes us. The ventral respiratory group is not in the solitary tract nucleus. This is in the nucleus ambiguous. Motor is what we should be thinking there. So these are going to be connected with the lower motor neurons that are going to control inspiration or expiration. And they're also antagonizing the activity of one another. We break the ventral respiratory group into two parts, the rostral and caudal portions. Rostral being higher and caudal being a bit lower. The rostral portion, which is actually up closer to the level of the dorsal respiratory group on the rostral caudal axis. So the higher up one, inspiration. Similar function as the dorsal respiratory group. This rostral ventral respiratory group is stimulated by the dorsal respiratory group, so they both work together. It also inhibits the caudal ventral respiratory group, which is more concerned with exhalation. Same thing is true for our caudal ventral respiratory group. It inhibits the rostral ventral respiratory group. So we're either inhaling or exhaling. We're not trying to do both. By having these two inhibit one another, by having the neurons concerned with expiration inhibit those neurons concerned with inspiration, that allows us to do one or the other, not both. So these groups are coordinating their own activity, but of course we have higher structures that contribute to this as well. So that reciprocal inhibition allows us to either inhale or exhale. That's kind of nifty. The descending excitatory input to the lower motor neurons is what actually carries out inhalation or exhalation. Inhalation is an active process. You're creating a vacuum, air is flowing sometimes. Exhalation is usually passive. So for the most part, what our caudal ventral respiratory group neurons are going to do is just inhibit the inspiratory neurons, and that's enough. The tension on the diaphragm is going to allow for passive exhalation. Only in cases where you need a higher respiratory rate do you have forceful exhalation, and then we'll actually stimulate the lower motor neurons that are concerned with forcing out air so we can breathe at a very rapid rate and not just let it leak out. It will leak out, and that's fine at rest. Right now you're not forcefully exhaling, but if you were to get up, run around, then these neurons would kick in and start to excite the lower motor neurons. Otherwise, they just simply inhibit the inspiratory neurons. And so that creates a cyclic inspiration, expiration, inspiration, expiration. When I'm breathing in, I'm inhibiting my caudal ventral respiratory group. When I'm breathing out, the caudal respiratory group is inhibiting the rostral and the dorsal respiratory groups, so I don't inhale. So we started here in the dorsal respiratory group, right there at the nucleus of the solitary tract. So here's our sensory input right here. This is going to tell us what's the pH of our blood, what's the stretch on our lungs. Based on that, we're going to either stimulate our dorsal respiratory group or not. If there's a drop in pH and we need to inhale, we're going to stimulate the dorsal respiratory group, unless the lungs are already stretched. Presuming that's not the case, and we do stimulate our dorsal respiratory group, that's going to activate the rostral ventral respiratory group and inhibit the caudal ventral respiratory group. The rostral ventral respiratory group also inhibits the caudal ventral respiratory group, so we don't exhale. What they also do then is project downward and stimulate the lower motor neurons that allow for inhalation. We have a few other systems to consider here. because breathing is going to require us to not be swallowing. If we're inhaling, we can't be talking. So we're going to have this pontine respiratory group up here, and then this more local pattern generator, the Betzinger and pre-Betzinger complex. These are what's going to generate the rhythm for breathing. How quickly should I be transitioning from inhalation to exhalation? It depends on the excitatory drive to our pre-Betzinger complex, because that is going to be connected throughout our respiratory groups. Parts of it are going to stimulate inhalation, parts are going to stimulate exhalation. And the frequency with which it transitions between active and silent in the pre-Betzinger and Betzinger complex. So these are our pacemakers, essentially. How quickly am I breathing in, pre-Betzinger, breathing out, Betzinger? It depends on how excitable they are. If they're very excitable because of excitatory adrenergic input, for example, well, then there's going to be a rapid increase and then decrease because of those calcium-sensitive potassium channels. So we'll get a rapid increase and decrease in activity and a rapid transition from inhalation to exhalation. There are many different connected systems here. Here we have our motor outputs that are being stimulated, of course, by our rostral ventral respiratory group, dorsal respiratory group. The Betzinger and pre-Betzinger complex, these are your pacemakers. That's the only thing that you need to get out of this. This is to tell you there's a lot of things working together. See, what matters here to you is that we have pacemakers that will determine how quickly should these transitions be made in those respiratory groups. So someone up above them makes that decision, and that would be your Betzinger, pre-Betzinger complex. They then provide the appropriate excitatory and inhibitory input to inspiratory and expiratory neurons based on their level of excitability. If they're not as excitable, very slow transitions in their activity. They'll slowly ramp up here, so you'll slowly transition from exhalation to inhalation. They're going to set the timing. That's about all you need to know. They set that through connections with the actual respiratory groups in the medulla. The pontine respiratory group is going to integrate a variety of information from other areas of the nervous system. So your trigeminal nerve, for example. Am I chewing? Am I fixing to swallow? That's going to help me coordinate my breathing. So the pontine respiratory group, a little higher up, a little better connected with other activities that are going on. Of course, the trigeminal is going to sense movement of the jaw there. In the mesencephalic nucleus, it will project on down. For the pontine nuclei, also in the pons would be this pontine respiratory group. So here's another central pattern generator. Based on what I'm doing elsewhere with my mouth and throat, that's going to determine, is it okay to breathe? Present that.
Thank you.
So that's one part of the equation, breathing. The other thing we have to do is move blood around, of course. For the most part, the degree to which we move blood around is going to be based on pressure. And we can sense blood pressure with stretch receptors. We have them on the aorta, we have them on the carotid, cranial nerve 9 for the carotid, just like it is for the chemoreceptor. Vagus nerve is going to have stretch receptors on the aorta. So we can sense blood pressure based on the amount of stretch that these baro or pressure receptors are detecting. So when there's an increase in blood pressure, there's going to be increased activation of stretch receptors, and that's going to feed into, again, the nucleus of the solitary tract, but in this case, the baroreceptor nucleus. Colored blue right there. This is where your afferents arrive. So sensory input from the carotid and aorta is going to arrive at the nucleus of the solitary tract and the baroreceptor nucleus, so the pressure receptor nucleus. This is going to determine if there's an increase or decrease in blood pressure. If there's an increase, we have an increase in excitation coming in from cranial nerves 9 and 10. If we excite the baroreceptor nucleus, then we stimulate a parasympathetic response. This is going to involve two things, stimulating your parasympathetic neurons and inhibiting your sympathetic neurons. We'll go through if there's an increase in blood pressure, that's what I wrote out on the slide here. If there's a decrease, it's going to be the opposite. So if we have an increase, our baroreceptors are now more active. They stimulate neurons in the solitary tract nucleus. They then communicate with their targets. First, we'll cover the parasympathetic nervous system. So down there in the nucleus ambiguous, again, motor. So we're going to have output here. The output would be the vagus nerve. The vagus nerve is going to project. Here's our preganglionic neuron. It goes pretty much all the way to the heart. It's going to hit a very local postganglionic neuron, and that's going to dump acetylcholine onto our sinoatrial node, and also the sympathetic postganglionic efferents that are there. So we're going to decrease pacemaking, just like we did back in lecture 11. Same thing. We're going to decrease the level of cyclic AMP. We're going to decrease HCN conductance, slower pacemaking. If you're not pumping blood through as frequently, you're not going to have as high pressure in the system. We also inhibit the sympathetic fibers locally. So we're increasing parasympathetic input, decreasing sympathetic output at the heart. We're also going to decrease sympathetic output centrally as well. So output from the solitary tract nucleus is also going to inhibit sympathetic output. Not only do we stimulate the nucleus ambiguous here, but here we're going to be acting in local medullary nuclei here. So in the ventral lateral medulla, we have a rostral and caudal portion that are going to act as the pressor and depressor centers. So let's walk through this stepwise. We have an increase in blood pressure. Baroreceptors are active, stimulate the nucleus of the solitary tract. They stimulate neurons in the depressor center. Neurons in the caudal ventral lateral medulla, the depressor center, inhibit neurons in the pressor center. So, excite, inhibit. Because these neurons in the pressor center are adrenergic neurons that stimulate sympathetic output. So what we've done is inhibit the activators of sympathetic output. We're inhibiting these. They don't excite as much. In the thoracic spinal cord, they decrease sympathetic output as a result. So out there peripherally or centrally, we're decreasing sympathetic output, so we decrease blood pressure. Sympathetic input would, of course, cause vasoconstriction there. That would increase blood pressure. We don't want that. We want to decrease it. We just sense an increase in blood pressure. If we have a decrease in blood pressure, the opposite occurs. We're not stimulating the nucleus of the solitary tract as much. We're not stimulating the depressor center. We're not inhibiting the pressor center. So that allows for excitation of sympathetic output and an increase in blood pressure through vasoconstriction. This gives you a fairly constant blood pressure throughout the day and as you change body position. Blood, like everything else, is going to be pulled down by gravity, and so when you change body position, blood pressure in different parts of your body are going to change. The baroreflex is going to allow your autonomic nervous system to automatically regulate your blood pressure in different parts of the body as they increase or decrease. So this keeps a fairly constant blood pressure throughout the day. So we can continue to move around enough blood so that we can get enough glucose and oxygen, pay the bill, and keep everything working. It also prevents dangerous increases in blood pressure. Any questions? Okay, let's talk about these, and then we'll move on to the next part. So the sleep-wake cycles that occur pretty much every day, if the day's going right. The wake is going to be stimulated by our ascending arousal system. And so this is going to be those more rostral neurons in the reticular formation that project upward, as opposed to did I say rostral? Yes, that's what I mean. As opposed to the more caudal neurons that are going to project downward. Now those are still going to be involved as well, because part of sleeping is not doing anything. And a big part of not doing anything is not exciting your lower motor neurons. But when you dream, you do things in your head, but hopefully not with your body, and most people don't. We have sleep atonia. Some folks will act out their dreams. This tends to occur in neurodegenerative states, such as Parkinson's, dementia with Lewy bodies, because they have loss of the inhibitory neurons that prevent motor output. So even when you're doing nothing, there's a whole lot going on. We'll start off by just covering the ascending arousal system. Notice we're fairly high up here in our brainstem. Down here, we're going to be dealing a lot more with descending tracts. But that's not what this is about. When we're talking about conscious arousal, what we're really talking about is the amount of cortical activity that we have. And that cortical activity is, of course, driven by sensory input, meaning we are aware of the things that we can see, hear, and feel. When you're asleep, sensory input doesn't make it to the cortex. It gets inhibited at the level of the thalamus, so that when people are talking, you don't hear it. Even though you still hear it here, that doesn't matter. Nothing matters until it makes it to your cortex. So all the sounds get filtered out at the level of the thalamus. And the thalamus is central to cortical activity. All roads lead through the thalamus. You're going to get to the cortex. And the thalamus is one of the targets of our ascending arousal system because it's so important for determining cortical activity. The thalamus and cortex are constantly communicating back and forth all day because the thalamus has input from all sensory modalities. It's more aware of what's going on right now than the cortex is. So there's a constant back and forth. That back and forth is facilitated through disinhibition. So the cholinergic neurons in the pons, and they're not highlighted here. Great. Okay. This is a useful cartoon, it turns out. So there's cholinergic neurons down here in the pons. There's a couple different populations of them. They will project upwards into the thalamus. Hey, there they are. They'll project upwards into the thalamus and inhibit inhibitory neurons. So there's one type of neuron that's going to ascend and allow for conscious awareness. It's going to facilitate cortical activity. And it does that by inhibiting the inhibitory neurons of the thalamus. So there's less inhibition at the level of the thalamus. So that when the cortex communicates to the thalamus, it talks back. It excites the cortex and allows it to be active. And that's what our consciousness is, cortical activity. Of course, coordinated cortical activity, not just noise. The noise comes in later on. The other population of cells is a lot more diverse. This would be all of the monoaminergic neurons, whether it be norepinephrine, serotonin, histamine. These project upward and more directly excite the cortex. Rather than stopping off at the thalamus and disinhibiting it, it projects up to the cortex and provides excitatory input to cortical neurons, for the most part. There's some exceptions, of course, but let's not think about those. Let's simplify our lives here. In general, we're exciting the cortex with monoaminergic neurons, and we're disinhibiting the thalamus with our cholinergic neurons. Also important here would be our hypothalamic neurons. These are going to be monoaminergic as well, so that tuberomammillary nucleus still releases histamine. This is still going to excite our cortex. And this is all going to be regulated by the lateral hypothalamus, both the tuberomammillary nucleus and the ascending arousal system. So those neurons that release orexin are going to stimulate both sets, the cholinergic neurons and the monoaminergic neurons. So if our lateral hypothalamus is active, we are conscious. This is our master regulator. Because these neurotransmitters, like histamine, for example, are going to stimulate cortical neurons. And that's what's going to make us feel awake. When we lose that during sleep, we don't feel awake. That excitatory input is lost. That disinhibition is lost. And the reticular nucleus of the thalamus is going to inhibit sensory input. So we don't hear things. We don't feel things when we're asleep, even though sounds are still happening and people can come touch us. As long as it's light, they usually don't wake up. It's because they've lost this this excitatory input. Here we can see a clear increase in the firing rate of neurons in response to histamine. Histamine is excitatory. That's still true. We've heard about this in previous lectures. This is just a reminder. Histamine is excitatory. Antihistamines will, of course, be inhibitory to our cortex and make us feel drowsy. What will also make us feel drowsy or awake is the hypothalamus. So we have our sleep-wake circuitry from lecture 12. This is all still the case. The master regulator is the suprachiasmatic nucleus down here near that optic chiasm. This will receive input from the retina about light levels. How light is it? More specifically, how much blue light is there around me? That's what's going to excite these. Based on light levels and the circadian regulation of gene expression, the suprachiasmatic nucleus is going to be active during the day and not so active during the night. The suprachiasmatic nucleus inhibits the subparaventricular zone, which normally inhibits the dorsal medial hypothalamus. Here's the next important stop. So when the suprachiasmatic nucleus is active, it disinhibits the dorsal medial hypothalamus, so it can be active. The dorsal medial hypothalamus is important to bring up because it controls the lateral hypothalamus, that master regulator, the source of our orexinergic neurons that stimulate the ascending arousal system. So if our lateral hypothalamus is active, we are awake. We stimulate the other monoaminergic and cholinergic neurons that then excite and disinhibit the cortex. The dorsal medial hypothalamus is going to directly activate and disinhibit the lateral hypothalamus. Still the case that this inhibition here is going to allow for those rapid flipping between wake and sleep. When the orexinergic neurons are active, which they will be during the day and not so much at night, but when they are active, they're going to act on GQ-coupled receptors and provide long-lived excitation of their targets, the monoaminergic and the cholinergic neurons, the ascending arousal system. Then they project upward, and we're awake. At nighttime, when the SCN is less active, now we're not inhibiting this subparaventricular zone. It inhibits the dorsal medial hypothalamus. The dorsal medial hypothalamus is now less active, so it's not exciting the lateral hypothalamus, and it's not inhibiting the ventrolateral preoptic area. Now this comes online and inhibits the lateral hypothalamus. No more orexin release, no more ascending arousal system, and we drift off to a peaceful slumber. Here we can see that orexin is indeed excitatory to those neurons in the ascending arousal system. So here we're looking at a histaminergic neuron in the tuberomammillary nucleus. Apply a little orexin, what happens to the firing rate? A marked increase. All these vertical lines would be an action potential, and here they're just piling on top of each other. If you zoom in and just have a look at the resting potential, you can see that there's a marked increase in the resting potential, putting it much closer to the threshold, which explains that increase in firing rate. So if you release orexin, you stimulate histaminergic neurons and other ascending arousal neurons. Those then stimulate the cortex and keep you awake. Yeah?
Where are the orexinergic neurons located again?
Lateral hypothalamus. Right here. At the end of our...
and they release orexin
yes.
to the cortex?
To the ascending arousal system. So we'll go back to here. So we were just right here. There will be some local projections of orexin, but it'll also head down here to the ascending arousal system and stimulate these neurons to then disinhibit the thalamus and excite the cortex. So here's our circadian generator. If it's day for us, orexin is released to stimulate the ascending arousal system. If it's night, no orexin release, no ascending arousal system. So that inhibition between our day circuits and our night circuits allows for only one to be active at a time. Because there's going to be feedback. Once we stimulate our ascending arousal system, this is going to contribute to further inhibition of the ventrolateral preoptic area. So when our lateral hypothalamus is active, it stimulates neurons that inhibit the ventrolateral preoptic area to further keep us awake. Once this starts to slip down, though, and decrease in activity, we're no longer providing that same inhibition to the ventrolateral preoptic area. We're also decreasing the inhibition from the dorsomedial hypothalamus because of the decrease in suprachiasmatic nucleus activity. As a result, once we start to have an increase in activity in the ventrolateral preoptic area, it's going to inhibit the orexinergic neurons as well as those neurons in the ascending arousal system. So we get a quick transition. Only one of these can be active. You are either awake or you are asleep, because they inhibit each other, just like inspiration and expiration. So this reciprocal inhibition, you'll see it all throughout the nervous system. When we have a choice to make between one or the other and we don't want both, the simplest solution is to make them inhibit each other. Once the balance tips a bit, they take it full tilt all the way. Once one of these systems wins out a little bit over the other, it fully wins. Once we start to excite our ascending arousal system, we then inhibit the ventrolateral preoptic area, which allows further excitation of the orexinergic neurons, which allows for further excitation of the ascending arousal system. So it creates a nice positive feedback loop. That circadian regulation of suprachiasmatic activity then shifts us over here. So we'll get quick transitions, awake, asleep, and they'll be fairly long-lived, because the circadian oscillation and suprachiasmatic nucleus activity is about a day. You'll get your increase and your decrease. Here we can see the effect of an anesthetic on the ventrolateral preoptic area. This is just to highlight to you how important this is in regulating consciousness. Isoflurane is an anesthetic that causes an immediate loss of consciousness. So you put a little bit of that on a rag, put it over the snout, mouse passes out. It doesn't take long. The reason that it doesn't take long is because of this flip-flop, this rapid switch that occurs. The isoflurane is going to stimulate neurons in the ventrolateral preoptic area. That's where they're recording from. There's a little neuron here, they're patched on, they put some isoflurane, doesn't take long, boom, it's off to the races. The ventrolateral preoptic neurons are going to inhibit the ascending arousal system, and the mouse passes out. This is, of course, inhibited by those monoaminergic neurons in the ascending arousal system. So if you dump a little norepinephrine, it inhibits it. So you get to flip-flop back and forth. Once we're asleep, the journey isn't over. We're going to continue through the night, transitioning between different phases of sleep. There's two broad distinctions: REM, non-REM. REM, slow wave, however you want to call them. During REM sleep, we have our dreams. During non-REM, we don't. And the transition between REM and non-REM is complicated and still being worked out, but we are aware that there are two populations of neurons that control this: REM on and REM off. Isn't it nice when they name them like that? REM on, of course, turns on REM. REM off turns it off, because guess what? They inhibit each other. No surprise there. That's the simplest way to have this system. When REM off neurons are active, they inhibit REM on. Vice versa. When your REM on is active, they inhibit REM off. And what the REM off neurons are going to do is just inhibit corticothalamic signaling so that we enter slow wave sleep. The REM on neurons in the pons, on the other hand, are actually going to facilitate cortical activity, but it won't be driven by conscious input. It's going to be driven by memories and noise. And those two things are going to create dreams that kind of make sense, but make no sense at all at the same time. Your memories can color your dreams, but it's still just driven by noise. It's not driven by external input. It's driven by input from the pons. That's it. So these REM-on pontine neurons are going to do a few things that create REM sleep. They're going to excite the cortex, and that will make your EEG activity high frequency, kind of like it is when you're awake. It's a little bit different, but it's nowhere near what you see in slow wave sleep. This is showing you just coordinated oscillations of cortical activity. It's not decorrelated, suggesting that there's any sort of perceptions there. We see that in REM sleep, and indeed we do have some perceptions. You can remember your dreams. You don't very well, but you can remember them, at least the latest ones in the night. There's a collection of different neurons that account for our REM-on neurons. Some of them are going to create dreams. What they'll do is, if they're down here in the pons, they ascend up to the thalamus, and then they stimulate the visual cortex. What's stimulating our visual cortex are these neurons in the pons. What the hell do they know? They're not getting input from your eyes, so sensory input has nothing to do with the cortical activity here. So it's noise. You'll dream about things that make no sense around you, and you'll have rapid transitions in what you're dreaming about because it's just junk and your memories. It'll also stimulate gaze centers, so you get rapid eye movements, hence REM. So those REM-on neurons are going to just non-specifically stimulate gaze centers, and your eyes will move around. That gives you your rapid eye movement. This gives you your dreams. And you don't act out those dreams because we inhibit our alpha lower motor neurons. We'll cycle back and forth between REM and non-REM. I don't care about that. I do care more about this. So whenever we have REM-on neurons active, they're going to stimulate glycinergic interneurons that then inhibit the motor neurons. So when we're in REM, we're going to have some cortical activity. We may very well have some motor output from upper motor neurons. We don't want that to actually cause movement though. So we head it off at the pass. We stimulate these inhibitory interneurons locally, and they inhibit the lower motor neurons. So even though you're dreaming that you're running or fighting somebody, you don't try to run and fight somebody. The loss of this regulation, the loss of inhibition, prevents sleepatonia, and people will act out their dreams. This can be very bad for the people who share the bed with them. In the middle of the night, they'll have violent movements. But normally we don't carry these out, unless there's some neurodegeneration or something like that. And then, of course, we inhibit REM off. And that's just your reciprocal inhibition at play. Do we have any questions?